alive. Oh, it's alive. It's alive. It's alive. It's alive.
Boom! What's up, everyone? Welcome to Simulation. I'm your host, Alan Sakian. Very excited to be talking about synthetic biology. We have Dr. Richie Kohman joining us on the show. Hello.
Hello.
Thank you so much for coming on.
Thank you.
Really excited, really excited. It was because we were at the Church Lab in Cambridge over January interviewing George Church that we ended up getting in touch.
That's right.
And now here you are on the show, and Richie's background is awesome. He is a senior research scientist and lead of the synthetic biology platform at the Wyss Institute for Biologically Inspired Engineering at Harvard University. He oversees all research conducted by the synthetic biology platform, including advances in nucleic acid synthesis and sequencing, neurotech, gene therapy, aging reversal, gene editing, organism recoding, in situ omics, organ engineering, stem cell therapy, extinct species resurrection, and all aspects relating to the intersection of synthetic biology and synthetic chemistry. Richie is also co-PI along with George Church at the Department of Genetics at Harvard Medical School of an IARPA-funded project to map synaptic connectivity in large brain volumes. And you can find the links below to richiekohman.com as well as the Wyss Institute page, his LinkedIn profile, and his Google Scholar page. All right, Richie, let's start things off with one of our favorite questions to ask our guests. What are your thoughts on the direction of our world?
Yeah, so I think from a scientific perspective, it's a pretty exciting time to be around, right? I mean, science just keeps progressing, the technology keeps getting better and better. If you think about the CRISPR craze, which I'm sure we'll get to in a little bit, that didn't exist 5, 6, or 10 years ago, right? That's a relatively new technology. And so what's going to happen in 5, 10 years from now, it's kind of an unimaginable thing. It could exponentially increase and get very exciting. It most certainly will be exciting. But on the flip side, if you think about, in terms of the United States and the world, maybe politically, it's kind of a scary time, I think, in humanity. Especially as a scientist, when you think about kind of the post-fact argumentless dynamic going on, that's kind of quite scary. So we'll see how this trend continues,
Yeah, the fact that we haven't had some of the cutting edge technologies even just a couple years ago, and now those are kind of opening up the edge of knowledge for us, for people to go and dig and see whatever they can discover with these new tools that are being made. That's huge, but then also, like you said, there's a lot of geopolitical things that we have to get through. Richie, let's hear about you from your journey. Where were you born?
I was born in the suburbs of Chicago.
Okay.
So, kind of about 30 minutes outside the city.
And how did you get hooked growing up in Chicago? How did you get hooked into synthetic biology? How did your interest in science form?
It's just something that was always there. I was always a technical person. I always was interested in science in all of my classes and things like that. So there's no question in my mind that I was going to do something of that nature. I mean, maybe something like architecture, engineering, but that was pretty much kind of always a thing. We ended up going to the Museum of Science and Industry a ton. It was kind of like a second home. I don't know if we went there more because I was interested in it, or I was interested in it because we went there more, but I remember that as a good childhood memory.
And then how about then venturing all the way into like Santa Clara University, where you did your BS in chemistry, all the way up to the U of Illinois. You went back.
That's right.
For the PhD. Tell us about this.
So for me, it just ended up being at that time a question of what kind of science I was going to do. And I gravitated towards chemistry because I think it seemed like a happy medium between the highly mathematical physics and the more sort of fact memorization-based biology. I kind of like both. And so went for the chemistry route, and then went to graduate school because I liked organic chemistry because it was the one subfield that popped out that had some sort of pragmatism. It was like a tool, very applied. And so I went into graduate school with that mindset, saying like, I'm going to be a synthetic chemist.
And then what were your like big aha moments during the bachelor's and during the PhD where you were just like, oh my gosh, this is exploding my mind?
Yeah, I had a big transition really early in graduate school where I think I had committed somewhat earlier to that subfield. And I went to a small undergrad, didn't have a graduate program, so I wasn't really aware of all the kind of science that was going on at the time. And what ended up happening was Francis Crick, the famous scientist, died in the early 2000s. And it was big news. I was reading his obituary. And of course I knew him as the co-discoverer of the structure of DNA, like most people know him for. But then I was reading about what he had done for like the last 30 years of his life, and I found out he was a prominent research scientist in the field of consciousness. And at that time, I think I didn't even know that people studied that sort of thing. Right? It didn't seem like a scientific pursuit. And so it really just kind of flipped a switch. And it opened up a door, and as a sub-hobby, I almost became interested I definitely became interested in neuroscience. It was completely, as a sub-hobby, I almost became interested in my career path at the time. And so it would be this thing where I would get really interested, and I would read up on it, and I'd try and cram the two worlds together, and I couldn't find a way to do it, and I'd kind of get depressed and drop it, and then go back to what I have to do, and then something else would happen, and I'd go back. And this is a series of things that happened throughout graduate school. You know, the consciousness stuff is like a really basic kind of philosophical science questions. I mean, the science questions are philosophical, but like big questions in the universe. And then I remember seeing a lot of stuff on the brain-computer interfaces. It was a cover of Nature with this electrode array of this paralyzed person controlling things on the screen. And once again, I remember looking at that and then looking at what I was doing. And I was like, I'd much rather be doing this stuff over here. And so I'm trying to think of some way to make the paths kind of converge, and again, didn't happen, so I'd have to kind of scrap it and move on. And then there was a third moment when I found out about this technique called optogenetics.
Yes.
Right, where you'd be able to essentially stimulate the brain cells with light. So it's a combination of genetic techniques with hardware. Right, so you use a virus or a variety of genetic techniques to put these ion channels that basically move ions across the gradient with pulses of colored light, which is what turns in what cells do when they fire action potentials. So you can use all this genetic trickery to put these things in very specific places in the brain, and then just pulse it with light and get this amazing control. And I think that was kind of the straw that broke the camel's back. And I said, well, listen, I'm just not going to do this stuff anymore. I'm going to go and do this stuff. And so I decided to make like a pretty big switch. Because it was apparent I couldn't really bring the two worlds together. I've kind of making a successful trajectory into like biomaterials, tissue engineering, things that I found interesting. But I think I made a list in my head of my priorities, and I had this realization that like I'm not really going to settle for like the 15th thing on the list. I'm pretty much going to do the first thing on the list. That was, at least I felt relatively young at the time. I said I have a lot of time to go, so I'm just going to go for it. And was fortunate enough to have somebody hire me in a lab to do that sort of stuff, even though I had zero experience with it. So it worked out.
These are such, again, these reoccurring themes that happen from people that we sit down with, that we get the young museums that influence young kids that get into science, and then you wanted something that was translational. You wanted something that had like almost immediate impact.
Yeah. I mean, I think that's why a lot of people gravitate towards the brain sciences. I think that's why there's, the Brain Initiative. There's Brain Initiative sort of in a related context is that there's horrific brain diseases that don't have a cure. And all this is because of this massive complexity of the brain. And it's because it's so complex, it operates, and then you have your emergent property of your experiences, which is kind of the most amazing thing, right? I mean, that's, everything that you're doing or experiencing has some sort of material manifestation that goes on, which in itself is kind of crazy. But then you think of the opposite, that you can actually come from it externally and manipulate it, and presumably that's going to change your experience.
Yeah.
Right, so I mean, there's magnetic stimulation and things like that, and then there's these patients with deep brain stimulators, and you can read things in the literature of them being put in the wrong place and turning them on, and kind of bizarre effects happening.
Yeah, the fact that we can, not only that we feel consciousness from what is emerging from within our body, but then also that we can stimulate transcranially even and have an augmented experience. Wow.
Yeah.
Yeah.
And you have the whole history of like traumatic brain disease and injuries and all these very bizarre neuropsychiatric disorders to look at, right? So people get in a car accident, and then all this weird stuff just seems to happen.
Yeah.
Right? You wake up, you have this cohesive experience of the world, and you're like, all right, then what? It's just this one solid thing that's happening, and then something happens to somebody, and there's like a fragment that's offset, and you're like, I guess it's really just a million pieces working in concert. How does that even work? It's kind of amazing. And even the fact that, I guess, consciousness exists, right? That's kind of the amazing thing. Why does a series of things happen that creates them to be aware? Lots of things are complicated, like the TV's complicated, right? This building's complicated. A lot of biology is complicated, but something about the brain, specifically the human brain, where it's just connected somehow, and then it operates, and then you have the human experience.
Yeah.
And really, I mean, all society is the result of like these objects operating with some sort of agenda, and then you can look at like the history of the universe, and then like humanity comes up, and now you have these agents and like chaos ensues as a result. Right. The impact on the universe from the emergence of human brains is amazing.
Is it true that we don't know what 70% of the brain does? Still, I mean, I might have heard that a long time.
Well, no, I don't think. I mean, they used to say that you only use 20% of your brain, which is true, and that you only use 20% of your brain at a given time. So I think that got kind of coerced into like a pop culture idea that there's like this vacancy of like dark matter. I don't think it's quite like that. I mean, it's kind of on a crude scale mapped out. You know, people know what these things do crudely, but a cohesive theory of the brain does not yet exist.
Thank you.
Now, okay, so when you were finding yourself doing this like transition chemistry with neuroscience, was this kind of some of the work that you're doing with the postdoc at BU, as well as Harvard Medical School, the MIT Media Lab? Yeah, so tell us a little bit about that.
Yeah, so my postdoc was in the neuro field. So I got in a lab that did this optogenetics technique. And so for my postdoc, it was almost all mouse work. So I went from organic synthesis in a hood to like brain surgery in mice. And so I had a couple of different projects. One was a drug delivery to the brain, which is a huge problem, right? Things don't get in. You want to deliver a drug, there's all these barriers. So I worked with a surgeon at Harvard Medical School to develop like a surgical technique for that. And then we also had some like nanomaterial delivery agents that we did with that. And so my affiliation with MIT was also in that. So the lab that co-invented the optogenetics technique I was working with.
Okay. Yeah, and optogenetics for neuromodulation is beautiful, and I think it has a lot of potential for neuroaugmentation. It's a very exciting field.
Yep.
So then, what about then you kind of got into biomaterials engineering even like almost 4 years ago?
Yeah, I mean, so the thing about chemistry, despite the fact that I haven't really fully left it, it's a really, really useful tool to bring into biology, right? So if you can synthesize molecules, synthesize materials and polymers, you can apply that to just a tremendous amount of biological questions.
Yes.
So that always seems to work its way in because it just ends up being effective. So it's never gone. I think it's a good foundation to have people start off in chemistry, move into biology.
So chemistry enabled you to synthesize the materials that can then do things like expand tissues to make it easier to image it.
Yep, exactly. Yeah, so there's a technique called expansion microscopy, which we do a lot of. And that's really just a lot of polymer chemistry. You know, there's a lot of chemistry in there, and so it just was amazing how it was one of these things that kind of just found itself into the mix in terms of a tool that translated. You know?
Yeah, that's okay. And then give us maybe a couple more examples, and this will probably end up wandering us now into the Wyss Institute, because this is one of the things that Wyss also is doing, is making these synthesizing through chemistry these things like polymers that enable expansion microscopy to happen. So then now it's been almost 3 years. Yeah.
At the Wyss Institute, right.
At the Wyss Institute. And this is a big deal because Wyss Institute is biologically inspired engineering, and we have the first asset that lists all of the different things. Yeah. And then gene delivery, genome editing, cell engineering, protein engineering, nucleic acid synthesis, nucleic acid sequencing. I mean, this is just
the synthetic biology platform. So the Wyss Institute is broken up into platforms.
Yes.
Right, that are associated with some faculty. It's a really unique and exciting place because it's this, at least the platform and the whole institute is supposed to situate itself in between academia and industry. So it's supposed to take all the cool technology and then kind of transition it out into companies or licensing deals. So instead of things just kind of circulating in this academic space, is supposed to move them out into the world.
I love that.
Right, so it's a very applied place. And so another thing that they have is full-time research scientists that are tasked to do this, which is one of the things, it's part of the system that I'm in. Sorry, hit the mic. Because you just operate in a different way than if you're like a graduate student or postdoc in academia or even like a scientist in industry. So I always say it's kind of the best of both worlds, right? Because the stereotypes the good stereotypes of academia is that it's like novel, exciting things, right? You bring to life things that never existed before, you just pursue your ideas. The negative stereotype is that it's just a bunch of sort of irrelevant things that you can't communicate to the world. And then industry, of course, has scalability, right? You can make these huge companies with tons of people and you can crank out products, but at least if you can get the feeling like you're a cog in the wheel and you're just like part of this massive team, and companies don't have, especially big companies, don't have the ability to kind of move quickly because they're big. So here we can spend some time, we can try and troubleshoot these crazy ideas, and I think the best part is that we can take a huge amount of risk. So it's okay if we come up with an idea for something crazy, spend 3 years, it just bombs. I mean, you'd rather it not, but your career's not over. You just say, okay, well, let's try and push something else out through. So that's the appeal. If you're a grad student, a postdoc, you really have this time frame, right, where you have to like, I got 5 years or whatever, I got to get a couple papers, I got to get out the door. We don't have that pressure. We say, like, we can just focus on getting this stuff to work. Let's just do that.
So it's almost as though Wyss Institute and other, they're kind of like these scientific institutes that are able to run all the permutations of what could be successful science that is then applied in industry.
Yeah.
And then to be able to have that actually, it builds a more cohesive, I think, relationship between academia and industry. Yeah. Also, industry can kind of go off on its own with the products without talking to scientists too much as they iterate and stuff.
Yeah. It gets like escape velocity, and then they can kind of go. But if you're a big company, you know, obviously you're probably going to invest in basic research, but you shouldn't be your main thing. You probably are going to want to spend a very small time and effort pursuing really crazy things, whereas we should be spending almost all our time doing really crazy things.
Yeah, that's cool. So then synthetic biology platform is one of the platforms
that's right.
of Wyss.
Yep.
What are the other platforms?
There's a soft robotics one, there's diagnostic chips, like a lab on a chip one.
Cool.
Applied materials. I don't remember all the names.
Nice.
I mean, I have my allegiance to
you have
my
your
platform.
allegiance. Yeah, totally.
I'm not as good at remembering the other ones.
So chips, robotics. I mean, this Wyss ventures into other. Cool. So, all right, within synthetic biology, maybe teach us about like this idea of biologically inspired engineering with synthetic biology. Yeah, wander us down.
Sure. I mean, all the platforms have an engineering component involved. And I always just imagine that as just having a very applied focus. But there's also, I mean, there generally are a lot of engineers, a lot of people that do hardware development, a lot of microfluidics, a lot of things like that, because again, we're aiming to build something that can scale and do a lot of these tasks. So synthetic biology is broad. What we do is huge. I mean, this list is pretty big. It's ridiculously big. But you can talk about synthesis and sequencing, at least, is kind of the backbone of a lot of industries, right? So DNA, what biolab isn't doing or utilizing something relating to DNA. So there's a current market for DNA synthesis. It's done chemically. A lot of industries do that. And that has a limit in terms of the length you can make. So we're trying to push that limit, right? So if you go and order an oligo, it's generally about 200 base pairs long. So you can order that, and you have to do other ways to kind of get it longer. So we're trying to do it biologically, not chemically. So you'd be able to get 10 times that. That's one of the goals. Sequencing is one of the most successful technologies to come out of biotechnology ever. We're still trying to push that. The main thrust that we've done is actually in situ sequencing. So less effort towards what you'd classically think of as like a box on a bench that does sequencing, and more of like analyzing biological tissues and reading out sequences in there. Gene delivery and genome editing. I mean, genome editing with the CRISPR revolution is something that we do a lot of, and the delivery aspect is basically getting the proteins and nucleic acid there to do what it's supposed to do. So if it's a therapy, if you're going to do anything in humans, you have to deliver it. So that's a lot of viral engineering sometimes.
So out of the, let's start with the gene editing one. Okay, so this is, you mentioned this at the very beginning, just that just even a couple of years ago, this wasn't even roaring to the same degree that it is now.
Right.
Yeah, and then now it seems like we want to have genetic enhancements, we want to eradicate diseases, that these are potentially some of the most important things, especially when we have our children, that we could prevent ailments from developing. These are very important technologies.
Right.
Yeah.
And it's really just the ease of use of the tool is what it comes down to. So there used to be a variety of iterations of this genome editing technology that had been around for a while. The problem is they're just really hard to use. So there was these zinc finger nucleases, talons. The crux of a lot of this technology is that you need to cut DNA in very specific places and then control how it gets repaired. So that's a lot of the genome editing. So you used to be able to, well, you still can, but the previous iterations involved protein engineering. So you'd have some sequence that you'd want to target, and then you'd have to design the proteins that would recognize that. And that's a protein engineering task, and it takes time. And then as soon as Cas9 was invented, or I guess discovered, that was just a total game changer, because now it's a RNA, DNA recognition event. So you really just have to design the complement of the target that you want with this RNA, which is easy, because you know the sequence, so you just program the sequence. And then the enzyme just goes there and makes the cuts. So as soon as that happened, it was like the volcano erupting, right?
Yeah.
And so the Wyss Institute was one of the first labs to do the demonstration in eukaryotes. So it was like the initial invention was discovered like in a tube, and then there was like kind of a tie for this demo, and we were one of those labs.
Yeah, and to have that be a part of the revolution where you actually get to see it become so much easier to make the edits is what's next? What else within the multi-billion year evolutionary process that we have yet to discover that we can basically take and apply to our desire to bioengineer our future. Yeah, there's so much.
Yeah, I think the way we're going with it is we're trying to refine the tool so we can multiplex it, basically. So if you have a cell and you want to make a cut somewhere, you can do that. There's some off-target effects, people are addressing that. But we want to make, like, let's say 1,000 edits or something, right? We can't do that right now. So it's really trying to push the tool so you can do that. So if you have a cell, you could presumably engineer it, so you change the code so it's completely invulnerable to like viral infections. Then you have completely like virally impervious cell lines that you can use, and that has industry applications because it's not going to get wiped out and you're not going to lose $1 million from all the hit you take, you know?
And then why would could we be potentially doing this at the stage of inception of the child? That way we don't even need to be injecting with all of the vaccines down the line. Could we be making the child immune to the smallpoxes and measles at the...
Yeah, I mean, I mean, there's a whole field of vaccines, which is exciting and trying to do that. You can also edit earlier and earlier. That's like a super hot button issue right now. There's a recent issue in China, very controversial, so everybody's very up in arms about doing any kind of like germline editing. But presumably, if you have...
Why is that controversial?
Because you pass, if you edit your germline, you pass on everything that you do to future generations. And especially if you do it, you're making changes for the future of somebody who doesn't have a choice on the matter, right?
But if it's to prevent suffering...
Right. I mean, there are instances, there's a slew of genetic disorders that are well characterized that are very genetically deterministic. So if you have this, then you're going to get this disease. I mean, most things don't fall into that category, but there are some. And so this is presumably a way to correct that. I think some opponents would say you can just screen that out, right? So you can sort of create several embryos, sequence them, and then just pick the other ones, pick the ones that don't have that genotype. But the proponents of doing the editing would say that you're creating more embryos, if you consider that a precious thing.
Yeah.
You're now creating more embryos that you're not going to take on to something.
Yeah.
And then there's a slippery slope argument about the designer babies that people are very wary about. I don't think there's anything, the current state of the field certainly cannot do this huge editing. I don't think we understand genetics enough to even be able to do that, and we don't have the tools to even be able to make those kinds of engineering tasks, but that's what a lot of people are saying right now.
And then, you know, out of everything that we're going to even continue discussing, I just want to let people know that this is just such a small fraction of what really is what you're doing and what you're because if you go look at Richie's Google Scholar page, I mean, there's so many publications on there, and so to actually be able to dig into the weeds of that science, people can probably just reach out to you if they want to dig deeper into the weeds of the science, and we'll try to do our best throughout the conversation. But there's so many things that these apply to. Okay, you brought up the nucleic acid synthesis and sequencing. So sequencing technologies, this Carlson curve that we've been following has just been incredible, that now it's down to less than $1,000 to do a whole genome sequencing from tens of hundreds of millions of dollars. Insane. So, but then the synthesis part I want to just quickly touch on. So what were you speaking about earlier when you were saying you were synthesizing nucleic acids?
So DNA and RNA, there's a lot of vendors, if you need to order a short sequence of DNA, you just put it, you fill out the form and order it, and they send it in the mail. And
they send you a synthesis of nucleic acids?
Right, yeah, a little tube, usually it's dried, maybe a milligram at most. But
a milligram of dried DNA or RNA?
That's right.
And just this can be ordered from the internet, just from a company, and
and it's made completely synthetically.
Made synthetically,
right? Which is I think it's kind of a cool thing. I think there's maybe an intrinsic concept of vitalism, like if it's a biological molecule, it has some sort of maybe biological essence to it, but it's not really true. If you look at DNA, it's a molecule like anything else, and if you make it in a non-biological fashion, and you look at the two side by side, they're indistinguishable, they'll operate indistinguishably. So if you look at the DNA synthesis, it's established chemistry, it's been around for decades now. And the precursors are similar, but they're all kind of optimized in a way to make the organic chemistry of it work. But the output comes off the instrument is still the exact same thing, it's just a piece of DNA. But it has limits synthetically, like, think of your cells replicating. You have all this DNA replication machinery, you're making millions and millions of base pairs of DNA. And then if you go to the current oligosynthesis technology, it's like 200, right? So you can't make long sequences effectively. Or I guess the yield drops, right? So as you get longer and longer, all the imperfections build up and you kind of get crap. So there's alternate methods to kind of combine these things and make longer ones, but that initial part still has that limit. So we're trying to break that limit.
And what is a nucleic acid made of when you make it synthetically? What material do you use to make it?
The precursors are still the monomers. It's just the ends are kind of optimized to fit the organic chemistry.
The monomers?
Right. So you have there's 4 bases in DNA, A, T, C, and G. Right. The bases are still there. They're still attached to a sugar, and then you have that phosphate, right? And so you basically have the 2 ends that are going to polymerize, and you just have chemical groups on each end that are going to fit the chemistry.
Okay.
So they'll just stack one on top of another in an iterative way, and then you could do some final ways to kind of knock the protecting groups off the areas you want and oxidize the things to get the backbone or the phosphates the right way, and then you just cleave it off, and then you have your oligo.
I literally submit a sequence that I want synthetically created, and then,
that's right.
Wow. Yeah, holy cow. Okay, so then, so in situ omics, so you're doing sequencing, and what are the cells here, and how are you sequencing them?
Yeah, so this video, or the still, which I'll have the video up now, this is our technology called FSSeq, or fluorescence in situ sequencing. And so next generation sequencing is very commonplace. Most of the techniques actually are imaging techniques. You don't really know because it's kind of what's under the hood of the instrument. And most people don't care, right? People want to sequence something, they use the instrument, it gives them the data, and they're not particularly interested in what's going on in the machine. But we are, because we're kind of cracking this thing open and trying to force it to happen on biological samples. So what's happening in a lot of these sequencers are that you're basically getting clusters of the DNA or RNA, or DNA that was once RNA, that you care about, and it's doing this iterative chemistry to make them light up and change colors. The colors tell you what the base is, right? And so we're doing the same thing, just inside the cell. So if you look, this is a cell, this is all the RNA, or was once the RNA. Each one of those spots represents an RNA molecule.
Can we pause it, Ron?
Yeah, go to the still.
All right.
I can loop it. Can we loop it?
Yeah, we can loop it. Let's loop it.
Yeah, so,
Okay, and just to give an idea of size, what is this size here that we're speaking of?
This is about 20 micrometers, right?
20 microns?
Yeah.
Wow. Okay, and that's millionth, billionth of a
20,000 nanometers, and
nanometers a billionth, I think.
it's a power of 10 off of a micron. So you're talking about 2 orders of magnitude off of millimeter scale.
Off a millimeter scale.
Yeah. Typical. This is a relatively standard cell culture size. So.
And this is multiple cells.
Each one of these with this bright spot represents the nucleus.
And what cell is this?
These, I believe, are HEC cells. So
HEX cells.
HEC, which is endothelial kidney cells, human endothelial kidney cells.
Okay.
And I think these were chosen. We could do it in a lot of different cells. I think these were chosen because maybe they're fibroblasts. Just an easy to culture cell for demo purposes. The next slides have the actual brain stuff. But for here, what you want to look at is the little spot, right? So that was once an RNA molecule. We do a bunch of chemistry to it.
Yeah, let's go back quick just so we can see
the still is easier.
Yeah, so the fluorescence in the cell. Yeah.
Go back so it's not animating.
Oh, so it's not animating. Okay, got it.
The still.
Got it,
yeah,
the
yeah.
still. Okay.
Here it is.
Yeah, right here. Yeah, so each one of these dots is the RNA.
Is the RNA.
Yeah. And
there's that much RNA.
There's actually a lot more than that.
There's a lot more than that.
But that is sort of a subsampling of it.
And orange and pink and green are different RNA, or is it just different fluorescence?
Different bases.
Different bases.
Oh, right, okay. So we want to know the 4 bases. There's 4 colors, 4 bases, and so we apply the chemistry, and it lights up a certain color. And I don't know offhand remember the colors, but let's just say magenta is A. So you'll see the magenta, you'll see that spot, you'll okay, that position is an A, and green might be a G. Okay, green is G, and then you'll iterate, and they'll change colors. And that's telling you literally the sequence of what that is. That's actually what's happening in a sequencer on the benchtop, or at least a lot of them. But it's happening in like a glass flow cell in a very controlled environment. So we're adopting what's happening there onto the actual cell itself.
Onto the cell culture.
Or the biological sample. We could do it in tissue
biological
slices.
Sample.
We do it in brain slices. We do it in cell culture.
So you can apply, what did you call in situ omics or sequencing, and
that's right.
And how are you getting them to fluoresce like that?
There's some chemistry where you basically you'll have an unknown, like this stretch of an unknown, and you put a primer. And there's a couple of different ways to do it, but I'll go over maybe the most straightforward way. Then you have a primer and you have a polymerase. The polymerase will just fill in. It'll just like take and it'll put the complement of what's there. But we have special phosphates, the A, T, C's, and G's that get polymerized in. There's special ones that are labeled with each of these colors. And they also have a blocking group that means it doesn't just keep going. It puts only 1 in and it stops. So it'll try to polymerize. It extends 1, it stops, snap a picture, and then you cut off the fluorophore and the blocking group, and you just repeat. So it'll keep going 1 by 1, and you just read off what's there, and hence you get the color change.
And this is a moment-to-moment change of color that you're reading through imaging it?
That's right.
And then you can then say that because it was green, then it was pink, then it was orange, then you're reading what the RNA...
Exactly. Yeah, you get the sequence.
You're sequencing.
Yeah.
Moment to moment, you can capture the images that fast
Well, you know,
Yeah.
this is there's some fluidic steps in between.
There's fluidics.
Right, right. So you run the reaction, and then you kind of wash everything out, snap a picture, and then you have to run the other reactions and wash everything out, and so you iterate, and then you do the image analysis. You can make a pretty video.
Whoa, and so then every single one is every single one of the bits that are there of RNA are different. They're different transcriptions of RNA.
It's possible to have two of the same RNA.
Yeah.
I mean, it all comes out in the sequencing.
Yeah.
I think it's unlikely, as you can tell just from the picture, that most of them are going to be different. But you can analyze it and compare it to the non-spatial version and get that data.
And then you being able to sequence in the cell culture instead of in the sequencer, the importance of that.
Yeah, so I mean, when you do, without doing it this way, you throw away all spatial information.
Spatial information.
So if you had, I don't know, tumor biopsy,
yeah.
If you want to get the transcriptomic or the RNA profile, you'd have to just grind it up and then put it in one of those instruments. You could just have this list that comes out. You don't actually know where it came from.
Yeah.
So you could detect something abnormal. You wouldn't know if that's half the cells or one cell, and even if you did, you wouldn't know what the spatial relationship is. In biology, it's all spatial relationships within complex tissues.
Yes.
So this technique allows you to just retain that and find out where it is exactly at the moment.
So you can actually see the spatial relationships, moment to moment. Cool, rather than just getting a list of the sequence when you grind it up and sequence it.
And so in this example here, we're capturing the endogenous signals, right? So that's the RNA that's just actually there. But we can do tricks to label things with DNA just so we can read it out in this fashion, right? So microscopy is a very powerful tool, but you're kind of limited by colors. So it's hard to look at more than like 4 or 5 things. But if each thing has a sequence associated with it, then you're basically looking at 4 to the n number of things, where n is the length.
Okay.
Right? So if you have like 50 things you want to look at, and you could kind of barcode them each with a sequence, then you just have to do a couple of rounds of this, and then you can see everything. Right? So it allows this huge multiplexing capacity, and that's really the crux of the neural project that I can talk about.
Yeah, let's do that. Let's dive into the neural project. Yeah. So this is the Brain Initiative, mapping synaptic connectivity in large brain volumes.
That's right.
Super interesting.
Yeah, so as everybody knows, the brain is just immensely complex, and the connectivity is really, really complicated. Each neuron, in the cortex at least, which is, this is a mouse brain, by the way, connects to about like 8,000 other other cells.
Yeah.
And there's millions and millions of cells. So the connectivity is extremely complicated, very hard to figure that out. And synapses are tiny, right? So you're talking about like 10 nanometer scale, and then you have macro scale objects of being the brain. So how do you actually dissect that out? How do you figure out and map all these connections? I'd say the current way of doing it is just to cut the brain into really, really thin sections, stay in it and look at everything. But that's like a huge brute force technique that you can imagine having some scaling problems, right? So we're trying to find new technology to overcome those scaling issues. So what we're doing is we're barcoding the cells in the synapses and then reading those out. So instead of, if the cells can be thought of as roads, instead of tracing all the roads, we're just inserting signposts where they intersect and then reading those out. And from there, we can make a reconstruction of where everything crosses. Not a perfect analogy, but it kind of gets the point. So we do the writing of the barcodes with a virus. So this is a mouse brain slice that had a viral injection, makes the cells green, and hence the cells. But each one of these cells actually now has a barcode, like a molecular identifier. And it's it's exactly 30 long. So each one has a different one. So within this context, we can identify 4 to the 30 different cells, which is way more than cells that are in that whole entire brain. Probably it's like way more than the number of sands on the planet, grains of sand. So it's like some astronomical number. So the scaling issue is kind of completely taken care of by that, right? And then we simply have to read them back out and establish the connectivity where the adjacent ones are. That makes sense? It's always a complicated project to explain.
It is, yeah, it is. Okay, so you're injecting the virus in order for them to fluoresce.
In order to give them that molecular identity.
To get a molecular identity.
Right, so if you think about it, if you do colors, again, these are all greens. So you say, well, I want to find out this cell versus that cell, I'll just make them a different color. You're going to hit 5, right? 5 colors, which is not going to get you very far with identifying.
Okay.
So I need to give this sort of the same kind of thing, an identifier that isn't color. That is just something else that I can read out.
Like a barcode.
Yeah, so I give that a nucleic acid identifier.
Okay.
And then I use our sequencing technology to tell me what that cell is. So this will be cell number 3002 based on its sequence.
Okay. Right. Okay, so you can not only uniquely identify the cells in the brain, but then you can also sequence them individually.
That's right. And I don't even have to know. So if I sequence right here and up here, and I see a spot that gives me a sequence here and here, I know it's the same cell. Right? So I don't have to sit there and trace this path. I could care less about that. So we can do long range in addition to short range.
And then do you follow that same protocol of the in situ omics where you're fluorescing them?
Yep. We do the same techniques on those cells.
And that's how you sequence them after you've identified, given them the identifiers.
Yeah. So if you go to the next slide.
Okay. Cool.
That's what this picture will be. So this is a static image of one of the imaging cycles. So the previous video kept changing colors.
Yeah.
This one's not changing colors. It's just thick. So it's as if you're scanning through.
Okay.
But you can see each one of these the colored dots is one of those identifiers. And so if it plays.
Okay, the video? Yeah, okay.
I guess the next slide. Right, you're scanning through, so you see all these projections and going.
So we're moving from the interior to the exterior?
Yeah, just through the slice, from the bottom to the top.
From the bottom to the top.
Right, you see all these colors.
Yeah.
If we were to iterate in a line or just take one slice, it would flash in the same way as that other video flashed. And I'd be able to say like, oh, this is cell, and I can list off the barcode.
Yeah, got it.
Right, I can say, and this right here, that one dot, I can tell you what that is. I can tell you what that sequence is. And then if we find that another dot somewhere else that has the same sequence, I know it's the exact same cell.
Okay. And then you're taking a three-dimensional. It's kind of like you're taking the camera from the base of the brain and you're moving it out towards the exterior of the brain.
Yep.
And this is what we're seeing pop up as the kind of the neuronal structures that are being shown, and then the colors are based on what cell, what the sequence of the cell is based on, is the color that we see.
Yeah, so the gray is just the location of the cell.
And what is white then? White seems to be the most.
That's where, just where the cell boundary is.
The cell boundary.
So what was green in the previous one is now white.
Oh.
Just to free up color space, right?
Oh, okay. Okay, that's the cell boundary.
Yeah, right. So this is like one large projection.
Yeah.
Right, and that's like the cell body.
Those are cell bodies.
Oh, that's where the cell body is.
Oh,
cell body, and these are like the long dendrites.
Dendrite. Yeah.
Can we take a question from the audience?
Yes. Yeah. What is?
Johan is a molecular biologist, and he wants to know what is your intentions? What are your intentions of doing gene editing? Did you answer that question already? Your intentions.
Gene editing. But we don't. Right now, we're developing gene editing technology, right? So we don't have. The projects currently aren't on the applications of it. We're trying to make the technology better, which is then useful to everybody else doing any kind of gene editing.
Thank you for chiming in, Johan.
We do have
Thanks for
a recoding project, again, that has to do with virus resistance, right? So if you can basically change all the codons in a cell to eliminate one of them, a virus won't be able to actually operate. So you'd be basically alter the genome so it can still make all the proteins it wants to, but it's kind of handicapped to make the ones you've removed, and then a virus is immune. So that would be an application. That's
okay. All right, and then let's get a little bit deeper into this. So then eventually what we want to do is we want to be able to map the entirety of brains.
Yeah. Right.
And then when you do that, then that gives you the ability to, what can we do with that?
So that gives you half the story.
Okay.
Right. So in biology, and especially in neuroscience, it's basically form and function, right? So you need anatomy, you need physiology. So I'd have to know what that neuron does, right? So and then you need some sort of behavior and to overlay with it. So this is actually in the visual cortex.
This is anatomy.
This is anatomy.
So you're getting
just
the
telling
anatomy.
me what's physically there.
Physically there, plus
it's not
you're
telling me
also-
anything about how it functions.
But you have barcoded and sequenced them. That's another thing. So anatomy plus barcoding and sequencing.
Right.
But
the
then-
sequencing is here to give the connectivity in the anatomy.
Yeah.
Right.
Okay.
But there's no physiology. There could be.
we can't tell from the sequencing what the physiology. Okay.
So from
if
this,
they're excitatory or inhibitory.
well, we
like
would
that?
be able to. We have ways to figure that out.
Okay.
But I don't know like the firing rate in response to a visual stimulus.
Oh. Okay.
So what you would do is you'd have the mouse and you'd do some sort of behavior. You know, if you look at the visual cortex, you show it something visual.
Yeah.
Right, and then you record the activity, and then afterwards you take the anatomy and then you overlay it.
Okay, so I will show images if we're doing a scan of the architecture of the visual cortex. I'd show an image, and then I would observe what happens as I'm showing the image to see what's happening physiologically.
That's right.
And then you could do that over and over again for the different areas of the brain to understand the physiologies that are occurring based on what stimuli you're inputting.
Yeah, in that case, then you're limited by technology for physiology. I mean, we do have one of these far out projects where we're trying to write in physiology into the cells.
How do you think that can happen?
So it's supposed to take the spike rate and encode it on a stretch of DNA so that we can read it later.
Oh,
so if you do that,
yeah.
Potentially you would have the whole brain encoding temporal information that we read out at a later time.
Yeah.
That's a little trickier.
Yeah. And that's a spike rate, and that means like you'd have multiple spike rates over time, and
that would certainly overcome the ability to record from many areas. You'd be if you had a transgenic mouse that had that operating, you would know every cell's operation within that window. So normally the two ways that people are looking at physiology are the two main ways. The classic way, we put an electrode in and you just record, right? And you could put in multiple electrodes, you could put electrode arrays, but that's still only really subsampling the brain. It's only a small fraction, right? And what you could also do is express a bunch of proteins that light up. So when the cell fires, it flashes. And you can put a camera and watch it. Now you're limited to that area of view where the camera is implanted. But if you could do this other technique, it's basically recording through the whole thing all the time. So you'd slice it up and that information would be written into the brain tissue. And then we would do something like this to read it out.
Oh, it gets written into the brain tissue, and then you read it out later.
Yeah.
Damn.
We're
writing it into the brain tissue? What are the long-term effects of that?
I mean, it would be in a mouse.
Yeah.
And it would be for some designated period of time for an experiment. It's one of our lofty goals, but it's not as far along as the anatomy.
Yeah.
Right. The physiology is. There's a time component to physiology that makes it a whole separate challenge.
Yeah, they come.
Right, this is like.
Yeah, the time component to the physiology makes it the harder challenge. Yeah.
So this anatomy, this is like a one time point in that mouse's
Correct.
life. Sack the mouse, and that's what the brain is, and now we're trying to analyze it.
Yeah.
Right.
Okay, so you need the anatomy and the physiology to make the whole connectome.
Yeah. Well, to get the connectome, connectome is anatomy.
It's just anatomy, but it's more important to also get the physiology.
Sure.
Yeah. Interesting. And have we done a connectome yet?
There, the other technique, the slice thin and map, is trying to scale up. So fruit fly brains, things like that.
We did get those.
Yeah. But
and that's like a fruit fly brain has what, like a couple
still
thousand?
Millions. I mean, there's
thousands
still
of neurons or what?
It should still be millions. I mean, but it's
millions. Wow.
It's still. I mean, think of a human brain and then think of a mouse brain. It's a significant size reduction. And a fly brain is even smaller than there. So
It's
there's still a lot you
millions.
can learn from fly brain, right?
Yeah,
So
yeah.
I mean, actually, there's a lot of conserved anatomy. The cortex, the wrinkly part of your brain, right, that you see, it's kind of crammed in there, but it's really a flat sheet. And if you were to look at just a chunk of cortex in a human, in a mouse, a monkey, they're really hard to tell the difference, actually. The general anatomy is similar, right? You have this structure called the cortical column that presumably has some general algorithm that it processes information with. And it's just repeated across the whole brain. So people are just trying to kind of deconstruct the cortical column. So if you could do that in the visual cortex of a mouse, it should at least have something to say about like prefrontal cortex in a human. So it's a general feature of the brain with cortex. So even that level of understanding is still a bit elusive in neuroscience.
And okay, now the brain initiative stuff's fascinating. I'm super excited for you guys to keep making moves there. What about on the stem cell therapy side of things, all the way to the reproductive technology side of things? I love this type of stuff. I don't know, something about stem cell therapy gets me really excited because it seems like it's one of the big keys to longevity.
Yeah, so it's thus far kind of a smaller component of the SynBio platform. But it may be something that is emerging, right? If we're trying to put resources into it, I think it would be great if we can push more stem cell research, and we could do more in terms of the reproductive biotechnology too, because that's kind of an exciting area. I know I've only recently kind of cracked open this field and discovered this field of in vitro gametogenesis. So I'm not sure if you've heard of this, but you can take a human cell and de-differentiate into a embryonic stem cell-like cell called iPS cell. That's kind of old news. That already won the Nobel Prize. That's being done all over the place. And the glory of that is that's your own cell, so it has your own DNA. So if you were to differentiate into some cell for cell therapy, it wouldn't have any kind of immune rejection. But what's currently not possible in humans is to make germ cells out of that. But a lot of work has been able to do that in animals like mice. So it's probably going to be possible that sometime in the future, you'd be able to take your cell, convert it to one of these other cells, and differentiate it into a sperm cell. So if you have people that have fertility issues or people that have injuries, I know a lot of cancer treatments for kids actually makes them infertile as a side effect. So there's people who grow up and they just simply can't have any kids. This would be a way to kind of bypass your own reproductive system to develop these cells, right? So if that were to happen, then it's somewhat of a cure for infertility, and can almost change like the human dynamic of sexual reproduction to some extent. And I mean, it's possible that you wouldn't even. I mean, it opens up the possibility of like two men or two women to be able to have a baby with their own genetic identity. So huge ramifications for a lot of that stuff in terms of effect on society.
How do we program all the dumb people to stop having babies?
Yeah,
How do we do that?
yeah, that's a different question. I don't know.
Sure is.
There are. I think it's a very important topic, though. It is. It's a very important topic. How do we maximize our collective abilities on this rock?
Yeah.
Yeah.
And in
Yeah.
terms of in terms of reproduction, even if you could just do egg cell development, right? I mean, a lot of women freeze their eggs and are unsuccessful. It really, I think that a big immediate effect is on women's rights and women's abilities in society, because basically the biology is not even in terms of its effect on reproduction with men and women. A lot of women feel this pressure to have kids at an earlier age because they feel like there's a clock ticking. If something like this could work, it can somewhat eliminate that clock. And if that clock gets eliminated, that can have a huge dynamic just in terms of society that could be really beneficial.
We could wait until whatever time we want to have children.
Right.
Yeah. With the healthiest embryos as
Right.
well. Yeah.
Yeah.
That's great. Yeah. Yep. Part of that also, that question that Ranj brought up, it's also interesting to think about what reproductive technologies and genetic engineering could do for all babies that are being birthed into the world. If we do figure out longitudinally what really good genetic edits are for intelligence or working memory or metabolism, that we could, then there would be the bar of intelligence just keeps moving up.
Yeah, I think the challenge is that when you look at human traits like that, it never falls into like 3 genes that if they're active, this is your IQ. It's not like a switch that gets flipped. Yeah. It's like these massively complex combinations of genes that are not well understood. And you really get this nurture-nature component where it's really difficult to parse out how much of one happens versus the other. It could be that you get some statistical effect that you can monitor, saying like, oh, people with, on average, people with these mutations or these genes are this much smarter. And then if you would do the edits, it might not actually end up that way because it's less of a nature component than you actually think it is. Could be better use of time to invest in, school system reform than gene editing reform, right?
As long as they educate people on being critical thinkers and not obedient workers. Talk to them about enhancing their intelligence, not having them learn and to repeat, and to program, to be creative, innovative, find a better way for all of us.
Sure, yeah. I mean, I've always been interested in the nurture-nature debate, as far as I can remember. And it's still an open-ended question in terms of, like, for a specific instance, how much of it is one versus the other. It's almost always both, and in an impossible combination to kind of untangle.
This is a critical point, is that when you have this potential of the seed of a child that is trying to unleash itself fully into the world, if it gets squashed by the economic, political, and social machinery and inhibits its full creative expression, versus if it gets all the tools that it needs, if it gets all its basic needs met, and that if it's able to fully creatively express itself. So then one of our goals with the show is to constantly be propagating conversation around the world and action around the world around how to make a better social fabric that enables that full creative expression for every seed, because then you don't get the instances where it just gets squashed by the system. Other thoughts quick on stem cells. I really think that there's just so much potential that people have been saying now around stem cells for longevity purposes.
Yeah. I mean, aging is another thing that obviously is interesting to us and a lot of people. It's a very hard thing to tackle because it's such a diffuse thing, right? It's just age. It's almost tough to even define what aging is and how to look at it, because it's basically what's happening all the time to everything in your body.
The loss of homeostatic capacity.
Yeah. So how do you have a treatment or a cure that can like attack all these things or fix all these things or change all these things? It's really difficult. So there are a lot of experiments that I've seen where you take like the young blood of a mouse and put it through an older mice.
Parabiosis?
Yeah, you can actually take 2 mice and surgically connect them in such a way that their circulatory systems are fused, and you kind of average out the phenotype, right? The older mouse gets better and the younger mouse gets older, so to speak. So,
or you just take the young liter of blood from the young and give it to the old,
right.
And that way they regenerate the blood. The young does, and
Yeah, so presumably you can imagine a cell therapy that creates a mimic of the young blood, and then you can infuse that, and presumably that would have an anti-aging effect.
And there are also very powerful cells in the bone marrow.
Yeah, that's where a lot of the blood-borne stem cells are.
Yeah. So. And so then to be able to extract that in a safe way and then deliver that to people along their aging as well. There's all different types of ways that this potentially the growth of the stem cells as well in its own bioreactors, and
there's all these effects of aging, like your genome is mostly fixed, right? Your DNA is not supposed to change, but when it replicates, it does, so it accumulates errors. And there's also the epigenetics, like the way it's read changes because you get these marks on it that accumulate over time. So one theory is that a lot of the aging is this epigenetic noise that builds up, like things, everything, as the cells differentiate and form tissues, you get these epigenetic markers that kind of get diffuse over time. And so if you could just sort of rewrite those, you can stall aging. And that's a totally different mechanism than the blood stuff. So you might have to have like 6 or 7 different therapies going in parallel to really kind of win the war.
Yeah. This is a cool point too, is being able to merge the different therapies together. Just retaining the 15-year-old's homeostatic capacity throughout life and being able to experience more, travel more, create more, have more relationships. Spend more time with the people that we love, watch how civilization flourishes. There's so many components to longevity that are exciting.
None of us really deserve to live beyond 30, so anyway, that's just my opinion. I should have been dead a long time ago.
And there's so many other thoughts around the work that can be done with synthetic biology that we didn't hit, the extinct species resurrection, how important that is to have a deeper sense of awe for what has happened on the planet in the past, and organ engineering, these types of things. So there's still a bunch of other topics in synthetic biology.
And even if you think about the reproductive biotechnology, which I was discussing with respect to human fertility, presumably that could help with the extinct species as well. If you have the genome and you understand at least enough of the reproduction, you might be able to recapitulate that and bring back species. I mean, that's far out from what's being done, but who knows? It's a possibility.
Yeah, I'm for sure ready to have like a mini T-Rex, just hanging out around the studio that is like playful, that like just nuzzles.
Yeah, we'll get over it. It's gone for a reason. And when we're extinct, I hope they just leave it at that.
Yeah.
Let's go home.
Yeah. Think about all of the cool, awe that could be expand upon. And like you said, with reproductive technology especially as well, it's like, what can we do to be able to give people back power to eradicate disease? There's so many cool things to do there. Augmentation, like you said, is really challenging. It's like hard to figure out exactly where that's at. Okay, let's ask a couple questions on the way out that we like asking the people that we feature on the show. So excited to see what you think. Do you think we're alone in the cosmos?
I'm the kind of person who doesn't speculate as much as if there's no data. I tend to not wonder as much. It's a big place. It seems likely. But do I believe it, or do I know it, or do I think a certain thing is almost like an irrelevant question unless there's some indication one way or the other. So unless we find it, unless it's there, then we just don't know. It's sort of cheap talk.
Do you think we're in a simulation?
I mean, it doesn't feel like it, but that is almost meaningless. That's kind of, it's almost like a circular question, because if it's so compelling of a simulation that you can't tell it's a simulation, then you wouldn't be able to know if you're in a simulation or not a simulation. So it's almost like, what's the point of?
I can tell it is a simulation. You just don't see it.
Yeah. So I mean, it doesn't. I don't think there's any reason to think that we are, but it would have to be a pretty compelling simulation. There's theories of consciousness that say it's all an illusion, right? And so I always have kind of a similar take on that. Like, well, if it's an illusion, then it's so compelling that we believe the illusion, then does it make a difference if it's an illusion or not?
Then what do you think is the most beautiful thing in the world?
Beautiful thing in the world? Well, I can have, I guess, 2 answers to that. I would say, I still think the ability of a piece of matter to operate in such a way that it becomes self-aware is pretty amazing. So that's maybe an objective scientific one, but I think I can go more corny on a personal level, and I could probably say my kids. Mm-hmm. I just had kids 10 months ago. So
10 months ago.
Yeah.
Nice.
So I can definitely go with that one.
Yeah. Going into this exponential technology age, I'm curious, and the geopolitical craziness also that we're entering with it. What would you say is an important skill for kids to know, including even your 10-month-old and whatnot?
I think it's definitely on how to get to the truth, how to know why something is real or not, how to evaluate information and make an important judgment on that, instead of just memorizing things and just like regurgitating facts. The ability to know why you know something, or at least defend it. So if somebody says something, you say, well, why do you believe that? To actually have an answer, because it seems like nowadays it doesn't even get to that. There's not that wonder of self-analyzing, right? And I mean that goes hand in hand with science. That's just that's kind of what science is. There's an experimental component to it, but it's kind of teaching you how to think. Yeah. So it'd be great if everybody did, engage more in the sciences and or even philosophy, but that seems like a hard angle to push for the majority of people because there hasn't been a big influx in that direction, and I'm not sure what would take that to change that in the future. But definitely, critical thinking skills. Right?
And there's that's huge. It's well said about what we need for the future. Yeah. Richie, this has been a really strong episode. I really appreciate you coming on and teaching us about synthetic biology.
Yeah, thanks for having me.
Huge thank you for coming on the show. Huge shout out to Ron Vargas for producing and directing. Thank you very much. We greatly appreciate it, Ronnie. And I'd love for everyone that watched, give us your thoughts in the comments below on the episode. Let us know what you're thinking on it. Go and have more conversations about synthetic biology with your friends, your families, your coworkers, people online on social media. Get talking about it. Check out the links below to richiekohman.com, as well as his Wyss Institute profile page, LinkedIn, Google Scholar page. Check out those links, everyone. And also support the artists, the entrepreneurs, the organizations around the world that you believe in your community. Support simulation. Our links are below. Help us grow and scale. And go and build the future. Execute your dreams into reality. We love you so much. Thank you for tuning in, and we will see you soon. Peace.